That's why I start before 3 o'clock.
Yeah, that's very
Uh,
fine. Yeah.
okay, sir. Welcome to all. All of you are welcome to UL Space Club webinar series session 24. We have reached our 24th session. Today we have an interesting topic. Throughout the history of throughout the history, the humanity has fought battles with the forces of nature. The race for survival became a race for prosperity and authority over one another. The field of biotechnology is playing an important role in this game of survival. So today US Space Club presents a webinar on biotechnology, an interesting topic, and we have a respected person, an Indian person, to talk on this topic. Sriram will introduce the speaker. So before that, I just want to give you a few instructions about the program. Throughout the program, you are requested to switch off your mic and camera. Otherwise, it will be an interruption to the program. You please switch off your mic and please switch off your camera. Uh, Otherwise, it will be an interruption to other participants to hear from the speaker. And I request all of you to pin the presentation screen of our speaker. Otherwise, you will lose the presentation of our speaker, and it won't be so great. So please don't please try to pin the presentation screen of our speaker, and please don't try to present your screen. Don't click the present now button. So please don't try to do that. And we have chat window here in the Google Meet. So please use the chat window in a good way. The unnecessary talks and unnecessary discussions are strictly prohibited in the chat window. So it's for our question and answer session. You can type the questions for the question and answer session that you would like to ask to our speaker in the chat window. Please type your questions in the chat window, and we will select the questions for the question and answer sessions. So don't try to have an unnecessary discussion in our chat window. So don't try to do that. And we have a question and answer session with our speaker after her presentation. So please be ready for that. And I request all of you to stay back in the Google Meet even after the question and answer session, because we have a feedback session. So please stay back in the Google Meet even after the program. So one more thing that I just want to remind you is our UL Space Club quiz. We will have a quiz after the our presentation. We will have a quiz. The link for the Google form of UL Space Club quiz 24 will be posted by our member Adil Krishna, and he will post in the chat window. So you can click the link for attend the quiz after the session. The link will be open only after the vote of thanks of the session. So please try to attend the quiz program conducted by UL Space Club. So please don't miss it. And these are the main instructions. Once more, please switch off your mic and camera, and please don't share your screen. So we have no time to make now. So I just want to invite my friend Sriram to give more introduction about the program. Sriram, you are welcome.
Okay, Renata, am I audible?
Yes, you are audible, Sriram. Go ahead.
Okay. A warm good afternoon to all of you. Thanks to Arnatan for providing the instructions. I cordially invite all the scientists, veterans, teachers, UL Space Club organizers, students, and all other participants to the 24th session of UL Space Club's webinar series. We, the UL Space Club, is a foundation under the umbrella of Uralungal Labour Cooperative Society, which stands for enriching and flourishing the scientific aptitude of students, especially in space field. Within the 3 years from the starting, we conducted many great offline events, that's before COVID. And from the beginning of this COVID pandemic era, we started this new venture called the webinar series, which is going extremely well for these past 7 months. Today, in the 24th session, we have Dr. Soniya Evie as our lead speaker on the topic, an introduction to biotechnology. She's a scientist from Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. It's so special to us that today we have a lady scientist as the lead speaker for the first time in the history of UL Space Club's webinar series. So it's my pleasure and privilege to welcome you, ma'am. And the topic is too important and significant because you may hear that the Royal Swedish Academy of Science has decided to award the Nobel Prize in Chemistry 2020 to two women, Emanuele Carpenter and from Max Planck University, and to Jennifer from University of California for the development of a method for genome editing. They have discovered one of gene technology's sharpest tools, the genetic scissors. Using these, researchers can change the DNA of animals, plants, and microorganisms with extremely high precision. This technology has had a revolutionary impact on the life sciences, is contributing to new cancer therapies, and may make the dream of curing inherited diseases come true. So the topic is very relevant, and please be attentive in the whole session. And after that, we have Varunatan, Abhinandan, and all core team members here to give you the instructions when it's necessary. And the moderator of the session, our all-in-all Shajisa, is here to control the session, and he will give the information about our respected speaker. Our chief mentor, E.K. Kutisa, the former director of ISRO, is here with us as always. Jairam sir and the whole webinar family, including the eminent scientists all over India, will be with us in this session too. UL Space Club student fellow, Master Bharat Srijit, will handle the Q&A session after the talk. And the Adil Krishna, another student fellow, who is also the streamer of our webinar, is here to express the vote of thanks after the session. So I think without any further delay, I would like to welcome the moderator, Shajil sir, to take over the session.
Thank you, Sriram. Now we are on the 24th session of UL webinar series, UL Space Club webinar series here in this pandemic. And this 24th session is so special because we got an eminent lady scientist from Kerala itself. I told it's a part of this not in a male-female ratio or any other aspect, but we are proud to be with a lady who is honored by our state and the nation. And we were in search for this kind of a personality for the past few months. We started this webinar 8 months back, and we couldn't get such a personality of this honor. And now we are so honored and proud to be with you, madam. Because you are part of this prestigious science institute in our state that is contributing a lot of things, and in the form of research and for the normal life situations and some other aspects also, sir. And biotechnology is the science in the ancient era that starts from our kitchen, from our food and the substances that we make from things, living and non-living things using living organisms. That was the content. The bread we eat and all the soft drinks everywhere, there is the biotechnology we are applying in the field of agriculture. Now, the biotechnology is everywhere. In our life, there is in our genetics, in our microbiology, in our cell biology, everywhere. Even in the case of evolution, also described in terms of biotechnology. That much of result is going on here. And I remember the first time I met our honorable guest, Soniya madam, because I just visited that institute in connection with an inquiry on the academic aspect when I working with the CRT for the textbook development program. But then when we made an inquiry there, we got a warm welcome there. From there, we felt the hospitality of that institution and this great lady in that institution. I remember 5 years back, and I could contact her only 1 month back to take part this kind of webinar with us. She very happily accepted our invitation, and he told me that I am so active with my works, but I am ready to talk with your students. And that enthusiasm, that concern on our students, that's why he came here, even from his very, very busy business and other official research and other aspects. So we will be ever thankful to you, madam, be with us. And let me introduce her, our guest, Dr. Soniya madam, officially. She is presently working as Scientist G in Academic Dean and presently residing at Thiruvananthapuram. And we are proud that she is a product of University of Calicut. She completed her MSc from University of Calicut and PhD from University of Kerala. And Chief Scientific Officer in Molecular Forensic and DNA Technologies, Regional Facility for DNA Fingerprinting, RFDF, and in Rajiv Gandhi Center for Biotechnology. And she is the Adjunct Professor, Region Center for Biotechnology, Faridabad. And a lot of honors and awards she already received. One is the Janaki Ammal National Women Bioscientist Award 2019, and elected fellow to the National Academy of Science, India, and elected fellow in the National Academy of Biological Sciences, elected fellow, Kerala Academy of Sciences. Best Women Scientist Award she received 2019-17 National Academy of Biological Sciences, and INSA DFG Award for International Collaborative Exchange Program, Max Planck Institute, Jena, Germany, Indian National Academy of Science, and a visiting scientist. And now she is a visiting professor in McGill University, Montreal, Canada, and she awarded with Young Scientist Award in Agriculture in 1999, a State Committee of Science, Technology and Environment, Government of Kerala. And she is also a recipient of Best Outstanding Thesis Award in the name of Jawaharlal Nehru in 2017, and a total of 19 Best Paper Awards. She is working there. And she is part of so many professional societies like American Society of Plant Biology, National Academy of Science, India, and National Academy of Biological Sciences, Kerala Academy of Science, Society of Applied Biotechnology, Indian Society of Cell Biology, Society for Plant Physiology and Biochemistry, Society for Biotechnologists, India. And she is in research guidance in some of other universities like University of Kerala, Manipal University, and Region Center for Biotechnology, Faridabad. And she's guiding a lot of projects there. And MSc MTech project, she already supervised 260 projects and research in her research coordination. She is the organizing secretary, EMBO, funded 4-day conferences, micro and metabolic regulations planned in Tirunelveli during in 2017. And she has made a lot of academic commitments in advisory board member in Kerala Agriculture University and board of studies member, Biotechnology, Calicut University, board of studies member, Computational Biology Informatics, Kerala University, Department of Biotechnology nominee, and Department of Biotechnology nominee in Biosafely Community Cochin University, and external expert in Institutional Biosafety Committee. And she is also a PhD examiner in different universities in India. And she is part of this organization for the few years and few decades, and she is contributing well to this prestigious institution in our state, that is Rajiv Gandhi Center of Biotechnology. And in our US Space Club, we selected this topic because in space we are conducting a lot of experiments, not only in the space science or in physics or chemistry, everything. We focus the survival of life on this earth. That's why that much importance is given to the experiments we already conduct in space. And that's why we transform these UL Space Club environment or ecosystem to a STEM point of view and to support all branches of science. And we are seeing no compartmentalization in the field of science. We are taking science in a whole, and our students are growing in that way. And by doing these and by taking part in this webinar, the students get an exposure to that great institution, Rajiv Gandhi Center for Biotechnology, that will be done by our guest of honor, Dr. E.V. Soniya, Madam, and that will be a great opportunity for our students. And like in all webinars, followed by this webinar, there will be a question and answer session. That will be the most attractive point of this program. And after that, our students will make some UL space quiz based on the topic already discussed here. And Mr. Varun, Bharat, and our Sriram is so curious and taking these funny, the very important and funny questions from this webinar that will be administered in the form of UL Space Club after this presentation. And we are so glad to be with you, madam. And we make a warm welcome you to this forum of UL Space Club again. And I welcome all the students, all the veteran scientists of ISRO and other organizations. And along with that, I want to add some more that Dr. Soniya is having a connection with ISRO. I think his husband was in part of ISRO or VSSC for a long time. So that's a connection with the UL Space Club and space organizations. And I, again, without wasting any time, I welcome our guest and our most respectful Madam Dr. E.B. Soniya to present his presentation. Welcome, Madam.
Thank you very much, Shaji sir, for introducing me very, very elaborately and also with very nice words. I'm really thankful to you, sir. And again, I know that Byron and Sriram also made very good introductory words. And I'm very much thankful to Kuti sir because for giving me and Shaji sir for giving me this opportunity to interact with all of you. Especially, I think I am first time getting a special audience like this because usually I go for presentation among the scientists or some conferences or something like. But this type of audience, I'm first time I'm getting. I know that it's a like from 8 to like degree standard students are there. So if I speak in that, I speak my, I will present my presentation in such a way that everybody can understand. And if there are any problem, you can also tell me that you can stop, make me to stop, and then you can ask also. I have no issue. And like Shaji sir introduced me, I am from Rajiv Gandhi Center for Biotechnology, and I am taking care of the research programs, mainly in the genetic engineering of plants. Like Shaji sir told, we are also doing a lot of genetic engineering work, like cloning, introducing the gene into the even plant, bacteria, and also making the transgenics, and also having like that I will later I will describe that in. Even now we started using the CRISPR where you can edit the genes. So that is one of the area which I am very much interested and working. At present, I have now the 6-7 students working as my PhD students, and postdoctoral 2 students are there with me. Apart from that, as Shaji sir introduced me, like I am also taking care of the human DNA fingerprinting facility of RGCB. And as you know, usually the fingerprinting facility is a service section, service offered by the Rajiv Gandhi Center for Biotechnology, and this is mainly taking care of the issues like, especially we are taking only the court cases, not private cases, and these court cases are mainly for paternity test and even crime and lot of things we are even like for example, if somebody wants to go outside India, they also need some help, like their identity and all these things. And especially the mass disasters recently happened in Kerala, like Oki disaster, and even the Pittingal firework tragedy happened in 2015. That time also we were very proactive and has done took lot of effort, and we could identify every person missing, and we could that way we are also serving the Kerala government in many aspects. Apart from that, we are also doing some wildlife forensics. I don't know how many of you are aware of the wildlife forensics. It's a we have the in a forest in Kerala, and we know that there are a lot of wildlife, and poaching is a major issue there. So again, that is a punishable offense. So for that also, they need a lot of they need support from us, that we will identify the species, whether it is wild or a so that way we will, once it is identified that it is a wild animal, it is punishable. So that type of work also we are doing for the forest department. And apart from that, we also did recently, I don't know how many of you read in the newspaper, because we have done some elephant DNA fingerprinting. What is that? Because we have a lot of captive elephants in Kerala. As you know, Kerala is very famous for the captive elephants. And നമ്മുടെ ഈ പൂരം അങ്ങനെയുള്ള കാര്യങ്ങൾക്കെല്ലാം we use lot of, and even the temple things, we use lot of elephants. And again, there is a big issue that especially there are a lot of misuse is happening in that also. So the forest department really wanted us to make a unique DNA fingerprint for each captive elephant. Okay, so this each captive elephant in Kerala is having a unique DNA fingerprint. Earlier it was used chips. Chips were used for that, and the problem in chip is it can be replaced with another chip, or this chip can be used even the elephant dies, it can be used for another elephant. So again, that ownership problems, a lot of issues are there. So to overcome all these issues, our forest department asked RGCB can you do a special fingerprint for, I think this is one of the first time in India, not only in India, even I don't know, world over, whether they have done such type of things, especially I think some tiger, some small work is done. Other than that, nowhere else is this first first time we are doing all these things. So this is another thing we have done from RGCB. And now I can come directly to my presentation. And I don't know, Shah sir, how can I share that presentation? Present now. Can you just tell me how can I do that? Usually, in our cases, we present now.
Shajil Mash.
How can I select my presentation? That is what I want to do.
Hello, Soniya ma'am. Do you have any difficulty right now?
Yeah, because I gave present now, but
Okay.
I am not able to.
Okay, ma'am. So if you present now, I think you'll be getting a small window where you get your entire screen, a window or a Chrome tab. So would you like to share your entire screen or any window or something?
Because the presentation is in the desktop. So which one I had to press now?
Okay, so you can click on a window, or maybe your entire screen, anything you want.
Okay, that I have pressed and I got it.
Okay, did you click on your entire screen or window?
This is entire one. Okay, share.
Okay, now you'll get something. You have to click on that, and then share will be enabled. Okay, ma'am, it is enabled.
Yeah, it's coming.
Okay, it's coming. Okay.
No, but I think.
I think, ma'am, you'll have to switch on that window. You have to open the window.
Okay. Oh, initially I had to open the window. Is it? Okay.
Yes.
Okay.
Let me do
Sonia, ma'am, you don't have to switch off the presentation. The window has to be on. That is all.
Oh, I have switched off. Okay. 1 minute. So it is there. Now, let me.
Okay, ma'am. So what happens when you share the screen is that whatever is being shown on your screen, that is going to be presented.
Okay. So now I had to. 1 minute.
Is the presentation coming there?
1 minute.
Okay.
Because some windows are opened in my.
Okay, ma'am.
Usually, some other applications we only 1 minute. Okay. So, present now. I'm in the enter screen.
Yes, sir. Yes, ma'am. Enter screen and select it inside that box and share, make it share. Okay, it has come. It has come. No problem.
No,
Then minimize that window. Take your slide, madam. Minimize this meet window and take your slide. That's enough. Your presentation is okay. Minimize this meet window and take your slide.
Can you see that presentation?
No, we are seeing the meet window, Google Meet window there. Please minimize that Google Meet window and take that presentation. That's required. Presentation is active now. You take that slideshow.
Okay. Okay. Now.
Yes, it has come. Okay, it has come. No problem.
Okay.
It has come.
Okay.
Please make it in a full screen. That's
Right.
better.
Sorry that I got little confused.
Okay, no problem. No problem, ma'am. Okay,
This
it's
is just
okay.
to introducing. So before I start, I will just have a discussion about what is biotechnology. And anyway, this is nothing but manipulation of living organisms. That we know that bacteria, even fungus, microbes, coming under microbes, and even plants, even like human tissues, all places we can do the manipulation through the genes. So usually we need to have a lot of knowledge in the genomics when you go for the especially the manipulation type of work, because we are usually introducing certain genes or replacing certain genes or making changes in the gene. All these things we are doing. So, and again, the importance of biotechnology, as you know, everywhere you know that biotechnology, you can do that way, this way, but how much it is useful to the human beings. And we know that we have a lot of physical disorders, and also we know that this biotechnology is nowadays, even that COVID cases, we are all waiting for the vaccine to come. So that type of thing. So in the medicine and even producing the vaccines and even energy production, even conservation, even the agriculture, everywhere we can use this biotechnology. And again, as you know that Government of India, how many of you know about what all things Government of India is doing? We have a program in biotechnology taking care of by Department of Biotechnology coming under the Ministry of Science and Technology. So RGCB is a part of Department of Biotechnology, and they are taking care of all the biotechnological work in India. And as I think Shajil sir told when he made the introductory speech, he told that we are using bread and all these things. These are all ancient, coming under the ancient biotechnology. Even our curd, all these things, because even though we were not knowing that what is happening there, we were using this biotechnology when I think very long back. So, but we never knew that this is like something like technology is involved. And later came the classical biotechnology. This is built on ancient biotechnology, and especially like fermentation. There comes the bread, making bread, all these things came, and producing the food in a different way, and also started like in medicine, some application of biotechnology came. And later in the modern biotechnology, we mainly use the genetic engineering. So it's really an engineering. That how it is engineering, because in the genetic engineering, what we will do is we usually introduce a gene of interest into the organism. It can be a plant, it can be a human being, it can be a microbe. So that for the introducing a gene into the system, you need a lot of engineering work. That is how we are introducing it. You cannot just like that introduce a gene into the organism, because you need to have a lot of elements which will help the gene to be expressed in that plant. Expression means if you wanted to introduce a gene for some, like for example, for an hair color or something, whatever, or a production of a vaccine. But that genes to express, you need certain things, like there should be a promoter type thing, there should be a terminator, there should be some enhancers, this type of things are needed. So this is basically we are engineering using the genes. Okay, and we will have different terminologies for different types of biotechnological applications. That is why this green biotechnology is coming. This is mainly for the agricultural process, and red biotechnology is for the medical process. White biotechnology is for the industrial biotechnology. Blue biotechnology is the term that is used to describe marine and aquatic applications. And all of you might have heard that about the bioinformatics. There is the application of computer science or computational biology that we now call it as a computational biology, because using this computer science into the biological system is now became a very important area and very interesting also, because then only that bioinformatics is a really a part of biotechnology. Without bioinformatics, you cannot do any biotechnology work. So I will, if somebody is interested, I can tell you also how it is helpful. Otherwise, I'll go through, then later you can also ask. So this bioinformatics is an interdisciplinary field. That means both it will address both the biological problem, okay, as well as computational techniques. You have to use the computational techniques to solve the biological problems. See, if you wanted to know a particular character is there in the, for example, I can tell you for a plant. If you are introducing a gene, or if you wanted to know that this particular plant is different from the other plant, how you will know? You can go for the genome analysis. So genome analysis, how can you do? You can maybe with machines only. We are first isolating the DNA. After isolating, what you will do? That DNA, how can you know that what all things are there? So you had to then, I don't know whether you are aware of the words like sequencing. Then you had to go for a sequencing. Then after sequencing, you get some like A, B, like A, T, C, G, like that, certain like alphabets only. From that, how can you identify that what is it code for? So for all these purposes, you need to need a computational help which will solve the problems, and you can. It also helps to tell us what it is code for and how you can change it and what all things are there in that. Lot of information you will get from the bioinformatics analysis. That is why separate departments are now coming for the bioinformatics. It should be very strong. Nowadays, I can tell you that even though we are telling a lot about bioinformatics, getting a good bioinformatician is very, very difficult. So we all, even though we can do all such type of biological work and analysis part, we are always suffering, and we will usually outsource this. So we will usually, that outsourcing happens outside India also many times. So what happens? Our data, everything will have a problem. So we had to have an indigenous, like we should have enough people in India. That is very, very much lacking now. And again, India has become a very attractive base as a cost of infrastructure is relatively lower compared to the other nations. Because we know that many foreign companies, the major problem is because the expense for the workforce and other things. But India, it is very cheap. Like typically, like IT companies, IT, that is the major attraction to India and China. Why? Because of the, especially the cheaper qualified workforce is available in India. So like that, multinational companies have now also started preferring India because of this. And you get good like people also here, and they don't have to pay that much that they are giving in outside India. So these are, and again, India produces enough qualified graduates every year. We have our system also very good that a lot of people are now learning like biotechnology, even bioinformatics, all these things. And now I think India is also sufficiently enough workforce for means addressing all these issues. And with it, again, we know that pharmaceutical companies, all this like drug discovery, all these things are very important actually. Now we have, India is having the capacity to do all these things. See? That is why we need to strengthen our biotechnology as well as this bioinformatics subjects, especially to the young students. And we know that again, the Department of Biotechnology is having the initiatives. They already giving funds to the all the departments to generate and to establish a good bioinformatics center also. So they will give money also. And again, as you know, like as I told you earlier, like IT sector, biotechnology is also coming like that. We are having now enough companies and even many multinationals, and they all need large number of people to work there. So I think that our next generation, I think students who completed their biotechnology and other things, they don't have to worry much because they will be well placed. That is what according to means, that is what I know now, because that much importance is there for biotechnology, because a lot of companies started this type of activities. Because even like I don't know how many of you know that biotechnology also having supporting a BIRAC. BIRAC means like supporting the industrial collaboration with research institutes. So their condition is that there should be something which can be taken into the industry. For example, if you wanted to produce a certain protein or certain which is having a lot of like economical importance, so you had to like research people will do the research part of and how to do. Finally, they have to transfer to the industries, and you have to show that this is producing the. And then it will, if it is a good product, then multinationals will take that. Even we have a patent section where we will patent that and we will transfer that technology to the industries or whoever is taking that. And also nowadays in our like profile also, people always ask how many technologies you transfer. That is a very important thing. Not like earlier only we can tell that I have published in that journal and that's enough, but not now. It is different. And wherever you apply for some like whatever it might, it can be a position, it can be a job, like another award or whatever, or for other fellowships. One of the questions is how many technologies you have transferred. So that is going to be very important now. And as you know, there are medical biotech segment and agricultural biotech segment and the pharmaceutical market. And at the present scenario of the COVID, we know that how much competition is among the companies, and everybody wants to market something which is useful. Like, for example, if somebody is using they will tell that their product is giving like immunity to the COVID and such way. So that way people are started producing many things, and even some like mask, a lot of things. So they are telling that some biotechnology aspect that we have watered this particular substrate which is indigenously produced, and it is having a lot of. So that way, I think this is very important. And also the agriculture sector, because agriculture sector actually we are also using biotechnology a lot, because one is of course making to increase the crop productivity. What are things we can do? And other than that crop productivity, we also need to have that like markers. Markers means to identify, differentiate among sometimes for a typical example is rice. You know that large number of varieties that are present for the rice. So if you wanted to like breed for a high yielding variety to a low yielding variety, you need to know that how much it is different. Whether how can you identify? Because sometimes if you see the phenotype, it will be the same for both the things. There will be changes, but certain cases I'm telling. So in that case, these markers will have great help, and they will help you to identify and even for our GI, geographical indication and all these indicators and all these things, even patent, you all need the genomic data and also the this type of markers. Then only they will also approve that this is belongs to a special group or it is something important than some other groups. So that type of things also agriculture is very important. And also I will tell you more things. Very again, we know that there are a lot of manufacturing industrial enzymes are so after that medical and agriculture, that industrial level also a lot of biotechnology things are happening, like producing enzymes in large amount and such type of things, like industrially important things. And the government of India realized the potential and benefit of this industry at a very early stage and formed the Department of Biotechnology that I have already told you, which is a central agency responsible for the policy promotion of. See, they are making the policies timely, and if it whether it can be a human, like I don't know how many of you know that we need to have a DNA database. So, and a lot of things. So we have to have the policy for each and everything. So DBT is take care of all such things and promotion of R&D. What type of research activities institute or should do? And again, a lot of programs they are launching. National cooperation. If you go to the site DBT or BST or whatever CSAR, then you can see that they have a lot of collaborative work with, I think, all the countries, almost US, UK, Germany, and not only that, with all the countries India is having already tied up, and lot of projects are there they are giving, and even fellows giving. So the students who are attending maybe in this program, they can go to the website of the DST, DBT, and even there are certain things like IUUSF, this type of sponsoring agent, like our government of India initiatives are there. They can go to their sites and see what all things are. Even for their PG courses, they will get fellowships, and even after the PG for the PhD they will get. Otherwise, also as an exchange program, there are a lot of programs going on. So all such things you can get from all such websites. Like if you go to like DST, DPT, all these things. And lot of financial support is giving even after PhD for the PDF program also they are taking, even in separate sectors like agriculture, this many fellowships they will give, even medical and even like microbial, such type of things are there. So that much funding is there for like biotechnology area. And again, the good regulatory framework has been set up for the approval of GM crops. We know that GM crops, that is genetically modified crops, are I think a topic of debate for I think more than 10 years onwards, because people are really afraid that genetically modified crops will create a lot of issues to the human being. So, so government of India, what they are doing is there is a regulatory committee. Different levels of regulatory committees are there, so they will take care about the biosafety levels and what are the rDNA products. rDNA means recombinant DNA products. That is the gene if you are introducing into the plant or whatever it is. They have to see that this gene will not create any issues to the human being who is consuming that, as well as for the environmental things. So all these things are already taking care of that Department of Biotechnology. And as you when you read my CV, it is written that I'm a member or a DBT nominee of the biosafety company. So biosafety committee, sorry. So it is mandatory that all the institute, universities, or any private companies who are doing the genetical modification should have a biosafety committee, and it is the duty of DBT nominee to see that any such type of any work such means illegal or whatever we can say, that any safety aspects is not taken care of. So they can stop the work. So it is a mandatory thing. Everyone should do that. So it's such an important thing. And again, the government of India decided some changes in the drugs and cosmetic acts. There are a lot of things are happening actually. And again, as I told you, the BIRAC is again the government of India association with the private industries. They are also giving a lot of financial support. Even the also startups. I don't know how many of you know that, because everybody doesn't want to go for higher and higher studies. So some people after their degree they wanted to do some like the startups. So even RGCB is having a like program that is in Kochi Infra Campus. There it is called as Bionize. It is a state central association. The state government is giving a lot of fund to the startups, and we will do facilitate it, everything, and also do like a lot of like research activities and support for the state government. So I think now more than 30 startups are there already. It's very interesting to see that different types of work is going on. Really interesting also. If somebody is getting some chance, you can also go and in the infra campus. What type of activities they are doing, all these things are there. So that government is also government of India is also supporting for that and giving fund also to even to the startups and entrepreneurs. And even government of Kerala is also giving money to this type of, because sometimes I will be in the committee, so I know what type of work they are doing. That should not be anything related to genetically modified. They can do any startup. It can be IT, it can be anything. So, and again, good regulatory frame has been said. Okay, that is another thing. So, as I told you, they have applications like in health sector or healthcare and other things, medical, crop production and agriculture, non-food, that is industrial uses of crops and other products, and finally the environmental issues. I cannot elaborately tell you each and every point because that much time we don't have. So I'll just go through everything. If you have any doubt later also you can ask me, or now also you can ask me. There is no issue. And this is I copied today, just to show you that these what are the applications of medical biology to have some idea. So we know that pharmacology is the gene therapy. I am sure that all might have heard about this word. Gene, what is gene therapy? And again, stem cells, especially in the cancer case, these stem cells is very important. And tissue engineering. If you wanted to have a particular organ, that's tissue engineering is there. So I think our biology is that much like giving lot of research activities actually. And this is gene therapy. Actually, this is a cut and paste thing, and so this is just to show you that what is gene therapy is a use of DNA as a pharmaceutical agent to treat. This is here also the role of DNA is coming. That is why gene therapy. Okay. I don't know how many of you know what is the difference between gene and the DNA. If you can also Google and find out what is the difference. And it derives its name from the idea that DNA can be used to supplement or alter genes within an individual cells as a therapy to treat disease. So actually, we can use the DNA. You can introduce, or you can remove, or you can even alter. If the gene is having some sequence information, A, T, C, G, C, G, you can at least remove one C from that. Then entirely different function will be there. Okay, and there are two types of gene therapy. One is somatic gene therapy and germline gene therapy. Somatic tissues means it is in the vegetative cells, and other thing is reproductive cells, embryos and other things. So I don't have to elaborately tell about the tissue engineering and other things. And coming to the agriculture, I will little bit more elaborate, because why I am giving you more details about the agriculture biotechnology is I know that most of you might have heard about the genetically modified crops, GM crops, all these things. So just to have what is happening in the GM crops, what is the problem, I will go through little more in the agricultural biotechnology. So this is the application of scientific technique to modify or improve plants, animals, or microorganisms to enhance their value. So usually we know that because of our genetic character, our behavior, our appearance, all these are happening. So changes in the gene can also alter the characteristics. So manipulating the genes, agricultural biotechnology are able to engineer the plants they desire down to the roots. Okay, we can introduce that. I can show you at the end of the talk that what all things we can include in the plant, or what all things we can change. So all these things are there. Okay. So agricultural biotechnology is the collection of scientific techniques to improve plant, animals, and microorganisms. See, this is the agricultural biotechnology is also applicable to the veterinary science. Okay, and agriculture and veterinary, both, it is separate in both. And also some like microorganisms also, because to increase the yield, a certain type of like soil microflora is needed. That microflora contains the bacteria and fungus. So that is also very important. So they have the solutions to increase the agricultural productivity, and also they wanted to enhance the breeders' ability to make improvements in crops and livestock, and also they want some improvements. Usually that is not possible with the traditional crossing. See, that is where the plant breeding also sometimes uh, faces problem, because in the plant breeding you need to spend several years, because crossing and going for 1 or 2 generations takes a lot of time, and to make a new like variety, the farmer has to wait 10 to 12 years or maximum 10 years. So that is not an easy thing for a farmer. So in that cases, if the genetic modification or GM technology have great help actually. So this is just an history. I am not going to the history. And genetic engineering is transferring of a DNA and developing including certain characteristics in the plant, and then that will be useful for the farmers, and for how to having the increased productivity. And also they need crop damage should be the minimum, and again the weeds should not be there, and the diseases also should be less. So and in our lab actually we are working on black pepper and some of the spices. There our major aim of the work is to identify the disease resistant genes, which because in the black pepper the major issue is like a disease called disease called root wilt or kick wilt, which is caused by a fungus called Phytophthora. So if you wanted to have, so it is a devastating disease for the black pepper, and actually it's an our export-oriented crop that takes lot of money. Actually, we lose lot of money because of this disease. So actually, government of India, not only this, and typically it's an example. There are other crops also. So government of India really wants to have an improved variety, or it should because we can especially, and again that it affects export also. If some fungal remains are there in the plant or the fruit, it will get rejected. So in order to do that, they are funding lot of money for the disease resistance for the agricultural crops. So the major strategy we are following is we had to identify a gene from the whole pool of, and again there is a big difference between human genome and plant genome. I don't know how many of you knew about the DNA. DNA means a group of genes and coding genes. We can tell it as a coding and non-coding. What is coding? Coding means the gene which codes for a protein. Hmm? If it is not coding, just like a junk DNA, we will tell, it has no function. That is what we were thinking earlier. And when it, so only the coding genes will have a product. So if you uh, find out a gene, especially the disease resistant gene, it will code for a protein. Okay. And later, so in the gene that contains is a mixture of coding and non-coding regions. So the coding regions will have the functional, they will be functional, and non-coding will be there is no function at all. So in the case of if you wanted to have a resistance for a particular plant, first you have to do is you have to identify the disease resistant gene from its wild variety or some other plant, or earlier in the genetically modified crops, they were also seeing a gene from like bacteria. There came the problem actually from bacteria, fungus, or virus. So then we will introduce that gene into the plant. And again, a lot of issues comes that again that gene will create a lot of issues. And again, so that is why people started working on the like their wild varieties. They are finding out the genes from their wild varieties. It's typically like a breeding, because in breeding, what you do is you do with a susceptible one and a resistant one, and after certain generations you will get the resistant one. But the advantage of genetically modified genetic modification is you don't have to wait that much time. Instead, you can clearly pick out the disease resistant gene from the wild variety, and directly you can use it, you can transfer to the plant, susceptible plant, so that it becomes resistant. So that is one of the method. And so genetic engineering is transferring a gene of interest into the plant. And molecular markers, I told you that to select the plants or animals that possess desirable gene. If you find certain marker, you can tell that this is having this quality. So that is what marker. Thus, breeding is more precise and efficient. That can be used for the molecular breeding. Then molecular diagnostics, you also have this parallelly with the medical diagnosis. In the plant also there is a molecular diagnosis, which you can find out whether it is like any diseases and how it can be using some certain like lateral flow assay methods, all these things. And there are also vaccines that is mainly for the veterinary, like livestock and mainly, and human beings also. Vaccines are produced. How the vaccines are produced, I don't know how many of you know. For example, hepatitis. Hepatitis is a serious disease, and we know that hepatitis B, C are there. B is like there is a easily curable and there's no issue. But for the C, it is not easy to cure because there is no treatment, real treatment for that. So what they will do is they wanted to have a vaccine. For making a vaccine, actually a part of virus, actually the most virulent part of a virus is like its surface antigen portion. So you had to isolate that particular surface antigen portion, that genes coding for that particular sequences coding for the surface antigen. You had to cut those portions from the virus and take it out, and again you had to use another bacterial system. Bacteria will be harmless and usually used for the biotechnology things, and you had to use that. You had to cut and paste in the bacterial system, and then you had to see that this bacteria is producing the surface antigen, enough surface antigen in the culture. If it is producing that type of things, you will develop into later vaccine, so that will not create any issue. Okay. And another important area is tissue culture. That I think almost everybody knows that what is tissue culture. The importance of, I don't know how much time it already. Shaji sir, I'm taking too much time.
No, madam. No, madam. It's enough. It's required there. And because all the 9th and 10th standard students want to hear about all these things, sir.
Okay. So this is a simple technique, but it's a very interesting technique because this tissue culture is the regeneration of plants. So there is a big difference between plant tissue and animal tissue. Only plant, why the difference is the plant tissue is having a quality like totipotency. What is totipotency? Totipotency is regeneration. If you have a small piece of tissue from a plant, It can be a leaf, it can be a stem, it can be any part of the plant. You can regenerate the full plant out of this tissue, whatever you are having. It can be an anther, it can be a like any tissue, any part of even a root piece. You can regenerate full plant out of this small tissue. That is the importance. Very interesting area. I have taken my PhD in very long back, 1995. That time it was a hot topic. So I worked on tissue culture. So we used to make plants. It's really interesting also because getting different types of plants from tissue culture, and it can be like using different explant. We will that small bit of tissue we will call it as an explant. So different tissues we are making the plant out of that. And again, there are a lot of differences. The method by which we are making the plants, if it is taken from the leaf piece. Uh, and sure, it will be, I don't know, diploid and haploid. How many of you know that? I'm not going to that area. Instead, I will tell that there are different types of tissue culture. If you keep a small tissue you need a medium. Medium means for that you need to include all the nutritional things which needed for the growth. So it can be it should contain a macronutrients, micronutrients, amino acids, and also it also needs like a gelling medium, to make the medium intact. So, and again, all the work should be done at the aseptic conditions. Aseptic conditions means it should not have any contact with outside like air. So you had to do it either in the bottle or in the tube, and you had to make it room aseptic, like clean and microbe free everything, and you need to have such a temperature monitor and humidity. So with all the controlled conditions, you can grow plants out of any tissue you can have. So for that, you had to standardize the media. Media means which are hormones, whether you had to add, and based on that, you will get different types of like plants. There are different types of methods like I don't know how many of you heard about the callus. Callus is nothing but like a unorganized mass of tissue where if you culture a tissue, leaf tissue, root tissue, whatever, it start dividing and finally it becomes a growth mass of tissue. Okay, so that type of things are there, and again there are different types of method like then each this tissue can regenerate into a full plant. So that many number of plants you will get, or directly sometimes you will get that if you like keep a small tissue of leaf, you can see that after 2 weeks small plants are coming from the leaf ends. And again, there are another type of tissue culture where once you get some callus or something, you can keep it in a suspension culture. Suspension culture means continuous shaking with the specific RPM under light. You start getting some embryo-like structures that is called somatic embryogenesis, and it will undergo like all the stages of the normal embryogenesis and finally develop into a full plant. So it's a very interesting. So we were using that for mainly getting disease-free plants, and also especially everybody might have heard about the banana tissue culture. In Kerala, we were having one in Kochi that for the banana they were exporting also. So true to type plants, we can get ഒരേ പോലത്തെ എല്ലാം it will be everything will be same. അതേപോലെ നമുക്ക് ലക്ഷക്കണക്കിന് പ്ലാന്റ്സ് ഉണ്ടാക്കാൻ പറ്റും. Because I worked 2 years in ഇവിടെ ചെയ്തു സെൻട്രൽ ട്യൂബർ ക്രോപ്സ് റിസർച്ച് ഇൻസ്റ്റിറ്റ്യൂട്ടിൽ ജോയിൻ ചെയ്തു after my PhD. അവിടെ ജോയിൻ ചെയ്തപ്പോൾ what happened that one of my work was they wanted, because I don't know how many of you know that കസാവയുടെ ഒരു വലിയ പ്രോബ്ലം എന്ന് പറഞ്ഞാൽ ബിക്കോസ് ഓഫ് ദ കസാവ മൊസൈക്ക് വൈറസ്. കസാവ എന്ന് പറയുന്നത് ലവർ ലൈക്ക് എന്താ പറയാ നമ്മുടെ മരച്ചീനി റൂട്ട് കർവ്. സോ ദിസ് കസാവ ഇസ് ഹാവിംഗ് ദ പ്രോബ്ലം ദാറ്റ് ഇറ്റ് have a major disease called cassava mosaic virus, which will always decrease its yield, affect the yield. So they wanted to, so when they plant in another place, they wanted that plants should be disease free, but getting disease free plant is very difficult. In that case, what you have to do is, and similar that sweet potato also. So the again the project was I think as a DST or Government of India project. There they wanted to supply every state like around 20,000 or 10,000 number of mm, disease-free plantlights. So to produce that, you can use the meristem culture. Meristem, I know that most of all the students will be knowing. Meristem is like an actively dividing part of a plant or living thing. So that especially plant, and this there is no vasculature, so that fungal or bacteria will not be there in that particular portion. Even though it is a very difficult to take the meristem, you have to dissect under microscope and culture. It's a difficult thing. But once everything is successful, you get disease-free plantlets. Then you can multiply that, and then you can supply to the whoever is needed, especially whether the states or the. Similarly, one time, our that time our minister, I think Jairam Ramesh, he came to Kerala and he wanted to meet all the black pepper the research workers, because they wanted to know whether it can be possible to get disease-free black pepper plantlets. Because even though we are even our home gardens also we are um, we are growing these black pepper vines. If they are like infected ones, you will not get enough yield, and that also creates a lot of issues. So that type of things are very much important. So there the application of tissue culture comes actually. And there are, but as I told you, when it is coming from the callus, there will be a lot of changes, and then that will be, of course, create different types of plants that will later. You can also use like breeding. You can use sometimes it will be good varieties also that can be also used. And this is just to show you that how the tissue culture. This is I think 2002 or something. This is the black pepper we have done in our lab. You can see large number of shoots are coming. This is the media I have told you, and how the plants are coming. You can see that. And finally, you can even transfer that into, this is what I told you, somatic embryo. You can see this is a small embryo coming directly from the explant, and you can see that later it is coming out, and you can see that first the roots are coming, then shoots are coming, then becomes a plant. This is how we do the racks, and this is everything should be done under the aseptic conditions. Okay, this is the laminar flow hood, and where we will do all our work and keep after that inside like this. And one more thing I will tell you and then stop, because just to tell you that what is like, we know that there are a lot of genetic engineers and other engineers, but I don't know how many of you know about the nature's genetic engineer. There is a bacteria called Agrobacterium tumefaciens. It's a gram-negative bacteria, and its advantage is like it can introduce its DNA portion into the plant. So usually that portion is called a tDNA, tDNA transfer DNA. That transfer DNA, normally this is causing a disease actually. So that region of the tDNA will go into the plant, it itself transfer and go and then insert, it will be integrated into the chromosome of the particular plant. And it started acting, producing certain like divisions of the cells, and later it becomes a crown, looks like a crown gall. So it is called crown gall diseases, and the causative organism is a Agrobacterium tumefaciens, is a gram-negative bacteria. So that time in 1902, people were thinking that maybe the same thing with cancer also happening. Cancer also, what is happening, like division is not able to control, continue to divide. So, but later found that both are really different, and this is a different, this is a bacteria, and that is also different. And then later, when we started working on genetically modified crops, people found a lot of like information about the agrobacterium, and they found that if you replace the tDNA portion with gene of our interest, you can use it for infecting the. This is the bacteria looks like. This is the disease in plants, and then this can be any gene of our interest can be introduced into the plants via this bacteria. Okay, so only that gene will be inserted, other portion will not be integrated. Only that portion of DNA which we wanted to integrate only will integrate. So this is the one of the best, the easiest method we are using for making the genetically modified crops. Okay, these are the methods how we will introduce the crop. And apart from the normal agrobacterial mediated, there is a instrument called gene gun. It's like that using the gene gun. It's like a gun. You can shoot it with the DNA, and that will go and it will break the cell wall and then get integrated into the chromosome. So that way also it can be integrated. That is what we call ballistic transformation. We had to use the DNA coated with the gold particles. We have one in our institute, and this is a more, I think, difficult thing, because how it is like genes do their job. Actually, first DNA will be transcribed into mRNA. I don't know how many can understand. DNA is, I told you, both coding and non-coding will be there. After the transcription, only the coding region will be there. After the coding, this one gene will start producing the protein synthesis, and finally that protein will be final product is protein. And there are structural genes and enzymes, and they will create product and product B. So usually the inserted gene, I told you, from the agrobacterial transformation can be done to unrelated plant or completely different species. All you might have heard about the BT cotton, because it was also an issue, because people were really worried about the gene which was used in that case, because it is a portion of gene from the bacterium. And then we introduced that into the cotton plant against the insecticide, so that no more insecticide is not needed to spray. So you can reduce the cost. Instead, the plant will have the protection against these insects. So we can use this. We can use this for increased resistance to pest, even raise tolerance to pesticides, increase market value. A lot of things are there. And again, if you wanted to have a good transformation, you need to have a plant tissue culture, and you also need to have transformation systems. There are a lot of things that I cannot explain now. It's each bullet is. I had to tell, I think, many things, and heterologous promoter system, reporter, and all these things. And there are two methods, like directly, that is what I told you, using the gene gun, indirectly through this bacteria. And so the major steps involved, you had to have a prepare a tissue for the transformation. Then using the agrobacterium or the ballistic gun, you introduce the DNA. Then using the plant tissue culture, you develop the shoots. And you can go for the field test like vaccine. In the plant also, you had to undergo different levels of testing for several years. You know, a vaccine will come out only after 10 years. Why? Because it has to undergo different levels of trial 1, trial 2, trial 3, like that. Plant also, when we are introducing a gene of interest into the plant, it has to undergo multi-locational, multi-centric, many different types of test, field test. And also there is a committee from the Department of Biotechnology or the Department Government of India. They will also see any variation, any problem, any toxicity, any allergenicity, all these things they will test. Then only it will come into like multiple sites, multiple years. This is how it looks like. This is our gene of interest, and you had to have a marker to whether it is coded or not. This is the starter portion. Without this, it will not gene will not be will not produce any protein. So this is very important. This and that, this is the termination part. This both should be there, and this coding gene also should be there. This is this is a small experiment we have done in our lab. This is the expression, as I told you, surface antigen in the plant. We can also tell it as an edible vaccine. If you express that in the plant and if you eat that particular fruit, you will get vaccination. So this was I have done in the 2003-4 that time period. This is we have isolated the surface antigen of the hepatitis B surface virus, and then we transfer that into agrobacterium. With the help of agrobacterium, we introduced into the plant, and we produced enough plants, and we saw whether that this expression is there in the fruit also. So this is the method we have followed, and the plants will be having the insertion of this hepatitis. This can be used as an edible vaccine, edible. And you might have heard about many things, golden dyes, which contain provitamin A, so that the deficiency of vitamin A can be reduced. You can introduce anything into the plant, and you can make out of that. This is another plant, and maybe two combinations. And again, if you wanted to introduce something, tomatoes with many varieties, colors, shapes, anything can be made out of that. And also, if you wanted to express certain things in the fruit, that also can be done. See, you can see that edible vaccine also can be produced in the corn or anything, especially if you have growth hormones, you can produce in the plant using the transgenic plants. And tearless onion you can make. See, usually when you cut the onion in the kitchen, what will happen? Tears will come. So you can also make tearless onion and such. And again, many things. And you can see that color, different colored, purple colored tomato, because of the high anthocyanin is very healthy also. So that type of things, and also different types of cauliflower, different colors, beautiful colors you can see. It can be orange, purple. That also using adding some flavonoids, not anything chemical, but it is natural, already available genes. Using that, you can produce that. So this is really healthy one. So that type of things can be done. And before I stop, I thought of giving you some activities connected to space also. And space biotechnology is also like government of, I think, all over the world, people are trying, and especially using the microgravity and space technologies, and what all things happening for these plants and how it can be utilized in future. That is also high. I think ISRO, I think faculties can tell you or staff can tell you more than me, but still this is what I know. And they were having a small collaboration with, as far as I know, Agricultural University, Toronto, Kerala, and I think they have started, but later some issue, I think they stopped. So the space breeding was initially involved in space breeding to investigate how the plants grow in space and producing food for the orbiting astronauts. And space breeding is a cutting-edge science that combines astronautics with agriculture and forestry science. That is what I told you. Biotechnology can use any type of like, can have any combination. So it refers to the technique of sending seeds or microorganisms into space on a recoverable satellite or in other recoverable spacecraft. Returning to Earth, you can see any mutations happen to the seeds or microorganisms, and what type of mutations happen, whether it is desirable or not, whether it is more usable, more user friendly, anything more is there, any difference is there. So that type of things you can study. And again, it has been discovered that spaceflight can increase mutation naturally, because in a different environment, the mutations can happen, and hundreds of times than experienced on Earth. Usually, the mutations in Earth is there, but when you in the space flight is more than hundreds. So mutation rate is by hundreds of times. That is what. And again, the seeds sent to space, 12% of seeds sent to space in satellites manifested mutations of some kind in they always found that there are a lot of mutations and that we had to be use it, and sometimes that will be the great mutations which can give you a very good yield or very nutritional quality, that type of things. And the Chinese Academy of Science has proved breeding seeds in space is expected to become the strong driving force to bring about high yield and high quality crops that are hard for ordinary breeding methods. See, we otherwise see there are a lot of limitations otherwise, so that limitations can be overcome by this type of things also. Sometimes these are all sudden changes. We can't predict that what will happen. So sometimes that will be helpful also. So Chinese Academy, last time I got an invitation in 2018 from them that they invited me for a talk in the Chinese Academy Center, and so I found that they are doing very good science also there, and they are all doing all such type of things. And the main factors of space environment affecting plant mutation is high energy ion radiation. This was the maybe the recent microgravity, space magnetic field, and ultra vacuum and other physical stresses. And scientists intend to the major goal of these experiments were, especially in the agriculture, is that whether we can, due to mutation, whether we will get higher yield and disease resistant varieties, otherwise not possible normally. Abiotic stress resistant plant varieties, biotic stress resistant plant varieties, and several other desirable traits. So you can see that there is a example just to show that. Okay, this is what the size of the god you can see, and this is also, this is because of mutation. And sowing seeds for the space. Some people tried this experiment. What will happen if you sow the seeds in the conditions, in the space, and that also sometimes there will be changes. And what happens in the artificial light in the that also is studied. And in conclusion, I think space-induced mutation technique is an effectively new way to develop new crop variety. But I never suggest that this is because it's not that easy and it's not that. But we can try that also. Possible to obtain rare mutants with economically important traits. That is possibility. And the research on applied basis of space-induced mutation technique needs to be strengthened. And the research on applied basis of space-induced mutation technique, space strengthened. And before I end my presentation, I just wanted to tell you that these are some of the top leading companies in the biotech sector. There are a lot of companies. Some of them I listed. That's all. Biocon, Serum Institute. Everybody is now know that because of the COVID vaccine thing, it is in the news all the time. And the GlaxoSmithKline, all these things are there. So, and again, what will be the chances if you study the biotechnology? Can be a biomedical engineer, and you can also a clinical laboratory technologist, and also can be I haven't even touched the forensic science. It's also a very important area. Forensic scientist and pharmacist and biological scientist. Of course, there are a lot of research activities need to be done. So there will be a lot of positions also, even in India and outside India. And again, medical scientists. See, once you enter into the biotechnology, there is no barrier. Whether you are studying plant science or animal science or microbiology, these are all in the technique is same for all the organism you study. So that is why it is very important. And thank you so much for the patient listening. I don't know whether I have conveyed whatever I really wanted to, and I don't know whether you understood also. Thank you very much.
Thank you, Sonia madam. You accomplished your task and duty very wonderfully. That's more than what we expected. You started the journey of your institution. From your institute, you made familiarized with this institute with us, and you started this talk with the ancient connection of this biotechnology. We start from even from our kitchen by making curd, and we started and we gone through the bioinformatics and all the other agrotech and genetic areas and biotechnology and agricultural biotechnology, genetic engineering, molecular markers, and molecular other chromosomes like that. And I am proud to say that when I visited your institution in 2015 for the purpose of making a curriculum for the students, and we get a lot of information, and still I keep with me the a book already given to me with some paper presentation that we used for that in making the content of the 10th standard textbook. In genetics, we have this structure of DNA, recombinant DNA technology, genetic engineering, gene therapy. Everything is included in the 10th standard textbook. And the information, the basis of the information we got from your institute, now that information passes all over the state. And we are proud to be say that you made an extensive talk on these, the content of 10th standard textbook, and all our students are well versed with the content, and you guided to them to the high realms or the high scope of this biotechnology. And it was interestingly, you told me that in the case of wildlife science, we use this Rajiv Gandhi Center of Biotechnology, and for some court cases and for identifying the parents like that, and all these are interested things. And I think the students still think that DNA is a long chain that they could see in a single view. That may be their view. Still, they are with us. I think during questions session, you will make this some more clarification on these things. And we are so interested here that the natural bioengineering, or there is no scope for any vector or any other techniques. Only a bacteria will do, the desired gene will be inserted to these things. And again, we get the scope of this biotechnology, the way we get wonderful shape and color in vegetables and in organisms. The different colored cauliflower and different shape in tomatoes are the results of the application of biotechnology. Now we are living in the world of biotechnology, and even the scope of mutation. It is interesting to share here the information that there was an astronaut called Scott Kelly who was in the space shuttle mission for more than 5 times, and after he come back to Earth, and his genetic code was compared with that of his twin brother in USA, it is found that his genetic code changed little bit. That means in space mutation is very high degree mutation is there. And here from Chinese information we understood that genetic mutation we can transform it to a good manner. We can make new varieties of high breeding or high yielding varieties of plants for our sake, and even we can fight against the diseases. A good information, and you made a wonderful journey of these biotechnology areas, and I think most of the students will be attracted to that. Some may get into these research areas like that. Today we have, the number is very few because there is a launch by ISRO. Most of our students are interested in ISRO launch, then they are enjoying that success of that launch. We also take part in that happiness of ISR, the great organization here. And you also mentioned the things about space and its space aspect in your talk. In that case, anyhow, our students will watch you and hear you through the YouTube channel. It's already made it live, and that's available for our students. They will use it as a resource reference for them. And as a biology teacher, I will also take my reference, and I have to transfer this video to some of other friends. So we'll be ever thankful for your wonderful talk, and you have done the way that what we intended, madam. We will be ever thankful to you on behalf of this UL Space Club. Before we go into this question and answer session, we have two eminent persons with us. One is E.K. Kutisa, our mentor of this UL Space Club, and one more person who is my deputy director of ISRO, K. Jairam sir. And I invite E.K. Kutisa to make his own reflection on this wonderful talk. Welcome, sir.
Thank you, Shajil Maharsh. It has been an excellent day for us. For the first time, we have got a highly accomplished women scientist. a person who has eminent research, scientific, and academic background, and who is working in Kerala's own important biotechnology center. She had given a very good roadmap of how biotechnology revolutionized the whole humanity. And in Indian scenario also she drew very well. 86 DBT, Department of Biotechnology, got established. Of course, I don't have much direct role in that, but I knew Dr. Manju Sharma, the first secretary there. We had a few meetings as a representative of ISRO, and I'm very fortunate that way to get a remote association with them. And TCMB in Hyderabad, Dr. P.M. Bhargava, the director, very highly accomplished person. I had dealings with him through Secretary DST at that time, MGK Menon. So like that, I have some bit of a connection with biotechnology. Of course, as a Bangalore man, I knew Kiran Majumdar Shah, the biocon chief. That way, biotechnology is a relatively new entrant, but is making very big strides and impact on humanity. So you have drawn a very nice picture and given a very good canvas to the students to know what is in store for them if they are wanting to enter into this new field. As you were mentioning, IT was attracting the people. Similarly, BT began attracting. It became a very huge success in terms of academic activity and research activity in India. So our students will be excited, and we are very fortunate to get this topic and to get yourself, madam. And I don't know how to thank you. I knew that today was going to be difficult for you, but somehow it was got managed very well. And I thank you from the deep of my heart for devoting your time for our students and us. Thank you very much. We hope to have interactions again with our students and in various other matters. Thank you very much again and again.
Thank you, sir. We want to hear some few words from.
One thing I forgot. ISRO's launch today, the very first launch of 2020, is successful. The health of the satellite is yet to be known. So it's that way also very happy day for us. Thank you.
Thank you, sir. We are sharing that happiness together. And again, I invite K. Jaram sir to make his few words of reflection on this talk. We are not getting you, sir. And the mic is not working.
But you can hear, sir.
Okay. No.
Yeah.
Okay.
Very good afternoon, madam. And it was really a privilege to be listening to you. The topic is very interesting, and Mr. Shajil and Kuti sir has captured the mood of the ULCCS children who are very actively listening to you. Thank you very much for taking us into this wonderful world of biotechnology, which is emerging, and also you have given us a clear picture of the infinite possibilities of plant engineering and how the food security and sustainable food availability for the entire human race is managed and made fruitful through the biotechnology. So also you have also shown the path for many of our aspiring students and this one, which way to progress, whether you go through the conventional means of earning money or you go through the enticing possibilities of research into plant and plant engineering. And especially I am personally very much involved and enticed by the tissue culture that you are presenting. Some of the tissue culture things, I'm a retired person, I am just doing some little bit of cultivation. So generally people get us this tissue banana, this Kindle Nendrapala and that. So some of the things I am trying out. So thank you very much for the very nice talk. And hopefully, I think Shajila and Kuti sir will rope in for much more interactions in the future. Thank you once again.
Thank you, Jairam sir.
Actually, sir, I was not prepared at all for today's talk, and I could only go through. Maybe he may be knowing that after coming back, I hardly got 10 to 15 minutes. By that time, I just conveyed whatever I actually wanted to. And again, like Kuti sir told about Manju Sharma, actually, she actually wanted to come here for the last April to have a meeting with the ISR because Sadish Dhawan's the centenary celebration, he wanted to start with the National Academy of Science India. So Manju Sharma asked me, why can't you conduct that in RTCB, Sonia? Because she wanted all the ISR officials to invite and to have a meeting here. But unfortunately, due to COVID, we cannot even do anything. So, so that I think stopped. And maybe in future, next year or something, once the COVID subside, we will think of doing again. Just like ISR connection, I just told that's all. Okay.
Thank you, madam.
Nice. So nice of you, madam. Your husband is an ex-ISRO, and Professor Anji Menon, who had a lot of things to do with the creation of DBT. He was engineering chairman of ISRO. So we have a lot of ISRO connections in the process. Thank you.
Yeah, that's
Thank you, madam. And we realized that the talk was not a prepared one, that flow from your heart. And your commitment to the profession. That was reflected here, and it was purely.
Just like whatever I came to my mind, I told you.
Yes.
That's all.
Yes, that was really the real thing we wanted here. That's why, and it was very coincidence with the topic already they learned, and they get a good idea about this topic. And that wonderful session. And the children are waiting there with some questions with you. Okay. And I hand over this session to Bharat Srijit. He's also a student fellow of this organization, and he's a higher secondary student. Bharat and Varun Sriram, they will handle these questions sessions. And children, get ready for these things. Okay, and welcome Soniya madam for this.
Thank you, Srijit.
So much, Shajil sir. Good afternoon, everybody. I hope I am visible, audible, and clear.
Yes.
First of all, hearty congratulations to all those ISRO people here with us today. Kuti sir, Jairam sir, and Padmanabhan sir. In fact, Padmanabhan sir likes to be addressed Pappan uncle. So congratulations, Pappan uncle. There is something quite intriguing today. Well, we are UL Space Club, and we are a space club, which is obvious. So we deal with subjects like space science, astronomy, black holes, rocket science, and all sorts of things. So why do we transition to a subject like biotechnology, which at first glance seems to be so different? Well, space science and biotechnology are quite very closely interrelated, considering the fact that plenty of experiments are being carried out in outer space with regards to biotechnology, especially in places like the International Space Station. But more than that, there are certain factors which make biotechnology a very important subject. Well, ma'am has told you of how we practiced biotechnology in the ancient ages when we made curd, bread, and even beer. And now we've transitioned into the manipulations of genes themselves. And in fact, we have come to a time where the humans themselves can be engineered is, well, a not very distant reality. And along with that comes plenty of problems with regards to sociology, ethics, and all sorts of things. So biotechnology, as a matter of fact, is a subject which encompasses all fields of science and even things like sociology, economics, and even politics. So UL Space Club understands the importance of this particular topic, and we are extremely happy and fortunate to be hearing from a person as distinguished as Dr. Soniya. And it is my honor, of course, to be handling the Q&A session. So the first question is from Eldo Sam Verghese, and his question to ma'am is that how do biodegradable plastics are made? Does it have any sort of relation with biotechnology?
Yes, of course, because see, as I told you, there are certain bacteria which can be used for like biodegradation, and there are certain enzymes which is acting for that. And if you actually know the which gene is producing this particular enzyme, you can means make such an organism which can be used for genetically engineer, and you can use it for the biodegradation. So that is one way, or other way is like you can also use certain bacteria which you can identify which is having the property of biodegradability. That also can be used both genetic through engineering as well as through otherwise also you can use. So a lot of work is going on in this regard, and there are a lot of new updates and how it can be used, and what are the bacteria, what are genetically engineered organisms which can use for this also. Even the oil spilling, we have heard about a lot, because in the sea you have a lot of oil spilling happens, and there are bacteria which can use for utilizing this oil, and because to make it pollution free. So you can bacteria, it is very easy for genetically engineering, not like plant or other bigger organisms. Bacteria, it is very easy.
Thank you, ma'am. Up next, we have two questions. Two of them are from UL Space Club itself. Two of them are student fellows of UL Space Club. And these questions seem to be kind of closely related. So the first question is from Abhiram TP. And he has asked, what are the chances of biotechnology in the future of space technology? And he has given special reference to interplanetary colonization, etc. And since I know Abhiram TP personally, I believe that he's a great fan of space related movies, and I bet that he might have watched the movie Martian, where they actually kind of practice this particular thing. And the next question is from Adil Krishna. He's also a UL Space Club fellow, and he has asked, ma'am, can you explain about the possibilities of micro propagation in space explorations? So my request to you, ma'am, is to address these questions together.
Yeah, I think both are possible. I never say that it is not possible. We have no words that it is not possible. So it is possible. But I think nobody have attempted until now. If you can do that in the here, it can be possible in the space also. But what is the difference, the environment difference from here as well as that you have to identify first, and then accordingly you have to supply whatever is needed for the growth of that plant. That you can artificially do that with the spacecraft, inside the spacecraft, and then accordingly you can do. Then second question was interplanetary. That I cannot say anything, but still my feeling is that future it will happen. That is what I know. Because see, never we thought of many things. Now what is happening? The potential of biotechnology is actually we can't even think of how much things we can do. So that can do wonders. Only thing is you had to understand about a lot about the basics, like that you should have thorough knowledge, like how you manipulate genes, what way it can be used, and all this is possible. So according to me, there is nothing impossible. So maybe it takes some more time and to understand the things. That is all. That's what my opinion. It may be true or may not be true.
Thank you, ma'am. We certainly do understand that biotechnology is a very vast field, and its possibilities are endless.
We never even exploited most of the possibilities. We only, I think, started. So that's what I'm telling. So there are a lot of possibilities. Even see, I can tell you if I actually that researchers has to identify like based on that problems. See why research is not going to that area, because if I am comfortable with particular area, I will always stick on to that area, and all my research activities will be based on that. So that's why sometimes funding agencies call for special calls like developing certain things. So maybe we feel that is something unusual, but there will be a special objective why they are calling that type of particular projects and why they are funding. So I think everything can be. That's why. So it can be possible. I never say it is not possible.
Absolutely, ma'am. In fact, you have been very much successful in getting the students here getting motivated into pursuing this particular field, and hopefully many of them will be pursuing this particular
Because
field.
I enjoy my science research a lot, I can tell you, because it's very, I think, give you happiness also, very much happiness than any other work. That is what my feeling. Actually, I wanted to work 24 hours, but it's not possible many times. But still, I'm telling that much things we can learn, and we can even more and more you do, you will get more ideas again how to go forward again. So like earlier we were not able to produce many things. Now we can, if I wanted to produce in a certain thing in a large scale, which is not even, so there are possibilities. I can tell you an example. I don't know how many of you know that you heard about the secondary metabolites. Huh? These secondary metabolites are the medicinal, giving medicinal properties to the plants. They are like a typical example is curcumin, maybe piperine. All these things are giving you like medicinal properties. That's why we are eating certain herbs for like curing certain diseases because of the secondary metabolites. So these secondary metabolites usually is produced in plants not for their thing, but they want their survival, especially when, because plants cannot move from one place to another place. Like we are running away. If something is coming, we can run it, but plants cannot move that way. So what they will do is they will produce certain things which will repel like their enemies. So these are very, but for human, I think mankind, it is very much useful as a as it is having medicinal properties. So what I wanted to tell you that this can be, and again, and it is not possible that all the time we had to go to the plant and get all the property, this medicinal like compounds. So in that case, one method is, that is what I can, pathway engineering. Pathway engineering is nothing but, if you know, because that alkaloids and these things are divided into alkaloids like, or terpenoids, or like many things, phenylpropanoids, there are a lot of classification. So if you know the pathway, what are the major enzymes involved in that, and you can use this use the genes, pathway genes which are producing particular like compound, that enzymes, you can clone that, and you can directionally clone that, and also you can express in the like yeast, and then you can using simple glucose, you can produce that particular product into outside, and you can scale up using biofermenter, all these things. So that way technology you can even it's very advanced. So that type of things we can do in biotechnology. Anything, that is what I'm telling. You can do a lot of things in biotechnology. I don't know the scope, how much I can't even think of. That is the at present what, that is what I can tell you. The scope is beyond our thinking actually. Any
Thank
more?
you, ma'am. Can we move on to the next question?
Yeah, sure.
Okay, ma'am. So the next question is from Manasa K. Krishnan, and she's asked, ma'am, what is the method used to name a new hybrid organism? I think she might have been referring to maybe the criteria, taxonomic criteria or something, while you name a new hybrid organism.
Not hybrid actually. Hybrid is usually giving the name the same, like only a hybrid variety, that anything you can give. See, if I otherwise, if you find out a new plant or anything, people name it based on sometimes if I am the first person So the genus with the Soniya, that way type of people name. And if it is a hybrid, usually hybrid will be based on if it is a tomato, Lycopsychum esculentatum, then you can maybe you can put any name. There is no issue actually. But the original name should be there, like Lycopsychum esculentatum. Then you can do that hybrid. You cannot change the name, but in the case of new things, you can do. But if you entirely different thing, then you can name it accordingly. But usually the naming only comes when you identify a new thing. Then only naming comes. There is no name until that time. You are producing a hybrid out of Lycopsychum. You had to name Lycopsychum. Then you can that way.
Thank you, ma'am. And also to Manasa, find yourself a new organism and give it your name.
Yeah, then you can name. She can also name. But genus name should be the related to that genus. Then the species, that name you can maybe Mansiana or something that she can give. Yeah, species that she can give. Yeah.
Okay, ma'am. So the next question is from another student fellow of UL Space Club, whom you've seen today already, and his name is Sriram D. So I would like to request him to please come forward and ask his question.
Yeah, sure.
Okay, Bharat, am I audible?
Yeah, I saw it.
How, ma'am, first of all, I would like to express my sincere gratitude that such a fascinating session it was. The presentation was so good, and I stick all the way in the presentation. And my question was actually about a thing that you mentioned just before, the medicinal values of plants. That how can we modify the plants using agriculture biotechnology for using it as medicines which had no side effects and which may cure the inherited diseases or cancer like that? What type of research is going on that area?
See, one thing I can tell you that we cannot say that a certain compound is not, if you're taking continuously, sometimes that also creates a problem. You cannot, but usually the products which we call it as secondary metabolites or medicinally important compounds are proved by research that it will maybe cure cancer, it may cure maybe some other disease. But I cannot, I can also say that we cannot say it doesn't have any side effect. Continuously, if you use something, it may have sometimes. That also, when you do research and publish, you have to tell that, you have to see whether the toxicity test, all these things. And in many of the drugs at present we are using in the cancer, we know that even though it is beneficial for curing cancer, it also have a lot of side effects. So to reduce that, there are a lot of work is going. That is why sometimes chemosensitization using some natural compounds, they will add sometimes curcumin, sometimes piperine. That also you have to find out to reduce the toxicity. Otherwise, we cannot 100% say that it doesn't have any toxic thing if you take continuously. That is one thing. Whatever it may be. നമ്മൾ പറയില്ലേ അമൃതമായാൽ അധികമായാൽ അമൃതവിഷം എന്ന് പറയില്ലേ, അത്രയേ ഉള്ളൂ. Do not take continuously anything. If you have certain problem, you can cure that using the particular thing. After that, better to stop that.
Thank you, ma'am.
Yeah.
Thank you, Sriram. In fact, I sense the possible emergence of a new discipline in science, maybe something like engineered Ayurveda. Well, maybe Sriram, you have an option. You could maybe pursue it in the future if you want to.
No. Ayurveda is basically plant now. So if you do some engineering in plants, that itself is engineered Ayurveda actually. Ayurveda 100% depend on medicinal plants actually. And again, there are a lot of differences in Ayurveda. I can tell you. See, there are in earlier times, not like now, people were using the original plants from their home garden or from their play field or whatever. Then they, that time only they will pluck the plant, and that also there is a specific time for plucking the plant. Certain you have to take it in the morning or in the afternoon. There are specific time. That time the Ayurveda practitioners tell you that you should pluck that plant at that time, and which part of the plant they will tell leaf, it can be root or whatever it may be. And then also tell that you never use any preservative things. Instead, we had to make the kashaya or whatever in their home only, and that also for a limited period. And there are a lot of changes happen in the Ayurveda now. So everything is having engineered Ayurveda means maybe you can also think of better preservative and better way to keep the ingredients again in the active form. All these things can be taken care of. And again, now there are methods by which we are also doing barcoding. If you send, especially for the export, you have to show that what are the things inside the lehya or kashaya or whatever maybe. That also can be done in the plan if it is plans.
Thank you, ma'am. Next, I would like to ask you a question myself. So it is based on CRISPR. So I actually became familiar with this particular term when I watched a YouTube video like a few years ago from Kurzgesagt. So I don't exactly know what CRISPR is, but what I understand is that it is something which is related to the defense mechanism of bacteria. And they say that it is in fact being applied in a lot of things which are being done in biotechnology. So, well, my question is that, well, we have immune systems of our own. So how special is the immune system of a bacteria? And considering the fact that maybe this particular technology is useful in making bacteria which are more resistant to bacteriophages and which can make yogurt and beer and all these kinds of things maybe a little bit more efficiently. But besides all these things, how can CRISPR be so important and so efficient in biotechnology as a whole in other aspects of biotechnology?
There are a lot of applications, because in plants nowadays people are using. Initially I was talking about the genetically modified crops using the agrobacterium mediated transformation and using so now that is going to replace with the CRISPR. You don't have, so that again gives you more regulatory freedom, because here if you are using agrobacterium, you have to undergo different biosafety levels, and it takes years and years, and I don't know when you can maybe in the field you can grow in the field. But in the using CRISPR, there is no regulatory issues at present, because we are only using the particular gene and replacing with whatever is not used in the plant or animal. Now it is used in all the fields, like agriculture we are using, and even medical field it is using, and even everywhere it is using. Human being also uses stem cell, it is very much used. So everywhere it is using.
Thank you, ma'am. So we have two more questions left today. The next question is from Navneet DS, and he also happens to be a US Space Club fellow. So he has asked, ma'am, what is the principle behind the RT-PCR test, which is used for COVID-19?
The RT-PCR is nothing. Well, PCR means the amplification of a particular portion of DNA to millions of times. So in the case of why we use RT-PCR, I can tell you that if it is a RNA virus or something like that, RNA cannot be amplified, so you have to make it a DNA. So cDNA we will tell it. And then again, why RT-PCR again give you that expression level. Expression means if you are infected with COVID, that expression level is very important. If the level is very low, it will not create any problem. So for that also this RT-PCR is very important. So usually in our lab, why we are using that is, we will get gene will be there in particular system, but gene may not be expressed. Why the difference between resistant and susceptible varieties? I can tell you. In the resistant variety, the expression of this particular gene will be very high, but the susceptible varieties, it will not be very expressed. So using RT-PCR, you can identify that the expression level, how much is the expression level, how many folds it is expressing. That is a very important criteria.
Thank you, ma'am. So we have one more question left for today, and that is from another US Space Club fellow, well, Master Varun K. Mugudan, whom you have also seen already today. So let me happily invite Varun to please ask the final question.
Sure, Varun, ask.
Thank you, Bharat, for inviting me to ask this question. Thank you, madam. It was a wonderful session. My question is based on your research, exactly. Madam, you are working on genetic engineering of plants, right? But what's your opinion about spreading knowledge about GM crops to Indian farmers? Because in Vision 2020, Kalam Sir has told that it's the most important revolution that we should face on in the era of 2020 to 2030. But still, our farmers doesn't know much about these GM crops and its applications, much about it. And one more question is there. And do you think that GM crops will lead to the extinction of our local plants?
No, Varun, that I can tell you, because otherwise also always people find the only means focus on the high yielding varieties. So naturally that will create issues to the low yielding but wild varieties. So that's why we have that gem blossom collections and all these things. We always keep the all the wild varieties, and if even though you are not using genetically modified system, or that is a different thing, you always depend on the high yielding things. So only those varieties will be more used. Now I have seen that there are a lot of varieties of rice also, lot of medicinal properties, lot of good qualities, but the yield is very low. So farmer doesn't want to grow such type of means plants in his field, because he need money actually. If it is very low yield, how he can survive with agriculture? So normally people always use the high yielding varieties, but the problem for the high yielding varieties is sometimes it is disease will be more. That is the only criteria. For that, they will use lot of insecticide, pesticide, because they also have to survive now. So again, about the genetically modified crops you asked, I 100% I never say that you can directly use anything without like. So that is why, as I told you, that we have the regulatory system in our country, and they were taking care of each and everything. I know that. I am also well aware that what all things we have to see before you release a variety, especially genetically modified. There are you had to do a lot of tests, multi-trial tests, multi-locational trials, and what are the difference and any problems it creates. After studying all these things, only we will release a new variety into the farmer's field. So I never find any problem for that, unless and otherwise people are worried about genes. I don't know why people are worried about genes. We are always, it is not a thing to worry, because gene will not create any issue unless and otherwise. See, we are not including anything means dangerous, anything dangerous. Only the high yielding variety, the same gene which is having in other system also. Only the level of expression will be little bit more. So that is why we are taking the same gene maybe from the high yielding variety. Only it's a actually shortcut of plant breeding, nothing else actually. And people were worried because earlier, because of using the antibiotic resistance, even though it is not a problem to humankind, but still, I can tell you that it's okay, it's a worry. So nowadays, it is not there actually. Nobody will use antibiotic resistance. So that type of things we modified a lot, and now it is a system which can be very well we can depend on also. I never say that this is bad or something. But anyway, here people has to be well informed what all things are there. And again, I that time it was very big debate. That times, Doordarshan and many kendras asked me um, whether you can. That time they were conducting lot of debates, and I heard that he was asking the person who is moderating the that discussion was asking a farmer that if you are given a poisonous high yielding variety and very low yielding but very healthy thing. See, everybody will say that other one, but why he's telling it is a poisonous? It is not poisonous actually. That is the feeling it is created among the public. That is the only problem. That should be changed, and also the person or the government also should give the every means details of that, any possibility of any problem, that also given. That transparency should be there. If it is there, then we can definitely think of that. Otherwise, I never say that it should be.
Thank you for answering my question. That's all about it. I enjoyed your session, ma'am. It was really wonderful, and it's an interesting topic, biotechnology. I don't want to waste time, so I would like to invite Bharat.
Thank you so much, ma'am. Thank you so much, Varnata. It has been a very beautiful session today. And, we have a very limited amount of time. So I have not been able to ask all the questions. So if there are any questions that has been left unanswered, we can discuss those questions in the social media platform that's UL Space Club provides. And its name is Cosmos. So if any student among us would like to join this particular WhatsApp group where you get all sorts of new updates of UL Space Club, where you can interact with other students with similar aptitude and ideas as yourself, and also take part in a lot of interesting discussions, you can join our WhatsApp group. Our contact numbers have been provided in the chat box, so you can message us via WhatsApp telling us your request, and we'll be adding you right away, which is also happens to be the outer layer of UL Space Club, so you can get into UL Space Club via Cosmos as well. So if there are any unanswered questions in Cosmos, I believe Dr. Soniya will be there for us to answer those questions as well. And we also have a quiz competition after this particular session. After the vote of thanks is delivered, we have the 24th edition of UL Space Quiz. So please make sure that you attend that as well. And there are many exciting prizes, including cash prize for the winners awaiting for you. So thank you so much. It has been a pleasure to be interacting with you, ma'am. Over to Shajil sir for the further
proceedings. Thank you, Bharat. We have been here for two hours. We are lucky to have Soniya ma'am, because there was another engagement, official engagement was there, and I don't know how could she manage. I don't know how could she manage that. Yesterday I was in very great tension, but we are ever thankful to you, and you opened a new gateway, because all these students were interested in physical world of science. Now they are a little bit interested in this biotechnology and the world of science. And what we are doing, we are breaking the walls of science, and we are seeing the science as a whole. And we are going to conclude. Before that, we have a great mathematician with us. Mathematics is part of science, and Padmanabhan sir, who was in ISRO and is a mathematics expert, and in the calculation, everything. നമ്മുടെ സ്ഫടിക സിനിമയ്ക്കകത്ത് പറഞ്ഞിട്ടുണ്ട് ഭൂഗോളത്തിന്റെ സ്പന്ദനം പോലും മാത്തമാറ്റിക്സിലാണ്. അപ്പൊ I invite Padmanabhan sir to make a reflection on this biological talk. Welcome, sir.
Hello, can you hear me?
Yes, sir. Okay.
എനിക്ക് ബയോടെക് കേൾക്കാമോ?
Okay, sir.
ബയോടെക്നോളജി ആയിട്ടുള്ള ബന്ധം എൻ്റെ മകള് മുഖാന്തരമാണ്. അവള് ബയോടെക്നോളജി പാസ്സായിട്ട് യെയിൽ യൂണിവേഴ്സിറ്റിയിൽ വർക്ക് ചെയ്തു. ഇപ്പോ മോൺസാൻഡോ ജനറ്റിക്കലി മോഡിഫൈഡ് സീഡ്സിലാണ് കുറെ വർക്ക് ചെയ്യുന്നത്. ഇറ്റ് വാസ് എ സബ്ജക്ട് ഓഫ് യുവർ ടോക്ക്. And she supports the view that we should switch over to genetically modified seeds because of the population increase and the number of hungry stomachs are increasing. And the BT itself has come from a bacteria which protects the seeds. And she says the BT has been injected into the cotton to protect it against the infection, so that pesticides need not be applied. So that's a big area, but our farmers have been told wrongly, and they are not in the. Yeah, that is what is happening. Second thing is nature itself doing a lot of biotechnology development. Species come and go. New species come, and we adopt to that. So whether the genetically modified seeds and food whether our body will acquire tomorrow, it is on the way. But one thing I tell the students, you are interested in space and astrophysics, black holes, which are so big an area, but biotechnology is much more bigger an area. So much of science is hidden there, which has been explored by you. And you have to feed the people in the coming generations. So there are a lot of work I should say a lot of work. And Madam has thrown a lot of light into many, many areas. And Madam, I'm thankful I also learned so much from you. Mathematics mathematics can help you model
Sure.
formation.
When you make the algorithms and other things
Exactly.
for the mathematics.
Yeah, genetics, mathematics is or genetics is integrated with mathematics now.
Actually, mathematics, that is the only.
Yes.
It has to be done. Yeah.
It has to be done. It is being done. It is being done. But only thing is, we should not genetically modify a human being, and there should be law to prevent that. Otherwise,
there are ശരിക്കും law ഉണ്ട് വളരെ issues ഉണ്ട് പക്ഷെ anybody who want to break ഒന്നും പറയാണ്ട് ചെയ്യുന്നവരുണ്ടാവും ഞാൻ പറഞ്ഞത് I never support അങ്ങനെ എല്ലാം ശരിയാണെന്ന് ഞാൻ പറഞ്ഞു certain things you have to take care.
So, my advice to the students are not only space is important, come down to the genes and DNAs and plants up to your own world. Your world is so wonderfully open there. Please do research there.
And again, sir, ഞാൻ ഒരു കാര്യം നോട്ട് ചെയ്തിട്ടുള്ള when you do the phylogenetic analysis from a bacterium to you can see certain genes completely അത് എല്ലാറ്റിലും ഉണ്ട് മനസ്സിലായില്ലേ അത് ബാക്ടീരിയ പിന്നെ ഇതില് വരെ ഹ്യൂമൻ ബീയിങ്ങിൽ വരെ ഒരേ ജീൻ ചില കാര്യങ്ങളില് ചിലതിനകത്ത് കുറച്ച് നമ്പർ ഓഫ് ഇത് കൂടുതലുണ്ടെന്നുള്ളതേ ഉള്ളൂ വളരെ ഇൻട്രസ്റ്റിംഗ് ആണ് ചില കാര്യങ്ങൾ നോക്കുമ്പോൾ എങ്ങനെയാണ് അതിനകത്ത് വന്നിരിക്കുന്നതെന്ന് പോലും നമുക്ക് മനസ്സിലാക്കാൻ പറ്റും ആ ലെവലിലാണ്
അല്ലെ നമ്മൾ ലാറ്റത്തിൽ നിന്ന് പ്രോട്ടോണും ഇപ്പൊ ബോസോൺ എത്തിയ മാതിരി ജീൻസിന്റെ ഉള്ളിലേക്ക് കടന്നു ചെല്ലുമ്പോൾ ദി വേൾഡ് ഈസ് സോ വണ്ടർഫുൾ
നമ്മൾ ഞാൻ ഈ പറയുന്നതൊന്നും ഒന്നും അല്ല ആക്ച്വലി എനിക്ക് പോലും വളരെ കുറച്ചൊരു ഭാഗം മാത്രമേ എനിക്ക് അറിയുന്നത് more things.
They can apply mathematics, statistics, physics, chemistry, biology, even electronics.
Chemistry, physics already there, like almost. Statistics is there. Everything statistics need to be there. And even, except maths maximum ഇഷ്ടംപോലെ ഉണ്ട് പക്ഷെ നമ്മളത്ര അപ്ലൈ ചെയ്ത് തുടങ്ങിയിട്ടാണല്ലോ ഒരുപാട് സ്ഥലത്ത് ചെയ്യുന്നുണ്ട് മാത്തമാറ്റിക്കൽ ഇതൊക്കെ ഉണ്ട്
എന്റെ മകള് പറഞ്ഞായിരുന്നു അവിടെ യെയിൽ യൂണിവേഴ്സിറ്റി ഒരു മാത്തമാറ്റിക്സ് പ്രൊഫസർ he has modeled mathematically and gave a 3-dimensional video on an embryo becoming a child, how it grows and up to what happens till the birth of the baby, as in a movie. Completely mathematically modeled, and mathematics can save in diverting or concentrating a part of the research in a proper way rather than going different ways.
നമ്മള് ഓരോ ഏരിയയും ഡിഫറെന്റ് ആയിട്ട് കണക്കാക്കി ബയോകെമിസ്ട്രി അത് മാത്രം ബോട്ടണി അങ്ങനെയല്ല എല്ലാം ഇന്റർഡിസിപ്ലിനറി ആണ് അങ്ങനെയൊന്നും ഇല്ല ആക്ച്വലി സയൻസിൽ
Absolutely. I am very much thankful to you. Very interesting. But I couldn't see your slides. I don't know. There was some problem with my. But I was listening.
Yes, that is available in our video, sir. That's there in YouTube. It's available, sir. I will send you the link now itself.
I am control. Thank you, madam. Thank you very much. Very interesting.
Thank you, sir.
Next we are, okay, sir. Next we are leaving to the vote of thanks session. I invite Kuti sir to declare our 25th webinar celebration like that on the next week. Welcome, sir.
ഷജിൽ മാഷ് ഞാൻ തന്നെ പറയണോ
sir, you have to speak it.
Okay. Anyway, ഇന്നത്തെ ഈ very rare occasion and madam has made it a very very excellent time for us, unforgettable time. ഈ ഡിസ്കഷനിലൊക്കെ വന്ന പോലെ BT ബയോടെക്നോളജി ലൈഫിൽ കൊണ്ടുവരുന്ന ഇൻഫ്ലുവൻസ് എൻഡ്ലെസ് ആണ്. നമ്മളുടെ ജനനം മുതൽ ഭക്ഷണം, ആരോഗ്യം, ഫാർമ, ഏത് മേഖല നോക്കിക്കഴിഞ്ഞാലും ഇത് വളരെ ഇമ്പോർട്ടന്റ് ആയിട്ടുള്ള ഒരു ഫീൽഡാണ്. അപ്പൊ കുട്ടികളിലേക്ക് എത്തിക്കാൻ പറ്റിയത് വളരെ സന്തോഷം. ഷജിൽ മാഷ് പറഞ്ഞ കാര്യം, നമ്മളെ അടുത്ത സാറ്റർഡേ 14th November ഈസ് ചിൽഡ്രൻസ് ഡേ. That happens to be the 25th webinar. അതൊരു വലിയൊരു മൈൽ സ്റ്റോൺ ആണ്. For us, it is a very, very significant day. So we are celebrating it as a 25th webinar and as Children's Day. So also we are ready with a website for UL Space Club that will also be inaugurated that day. All these celebration and inauguration will be done by Dr. M.K. Jairaj, our new Vice Chancellor of Calicut University, and that function will be an important one where the President of ULCC, Ramesh Palari sir will be there, and we will have other important dignitaries participating, like Professor Lijesh, HOD of Calicut University, etc. Details will follow. You will get the post on other things. So that is all about next week's program.
Thank you, sir.
Can I interrupt? Sir, I just want to mention that our quiz link is posted in the chat window. Please attend the quiz after the session. And our contact number for joining our WhatsApp group Cosmos has been given in the chat window. Please contact in the number if you want to join.
Okay.
That's all.
Thank you, Varun. And now we are going to the bottom side session. Before that, I will again make a request to Soniya ma'am that we are developing an ecosystem. That in that ecosystem, some few genius students are developing here. The reflection we already get the Varun, all these things. We want to nurture them again and again, and we want some support from your part by making familiarizing with some other scientists and some other areas and suggestion to this program, and we expect from your part, madam, and that's why that's our request, humble request from our part. And I invite Adil Krishna to express a vote of thanks to this today's session. Welcome,
Sir,
Adil.
am I audible, sir?
Yes, you speak.
Thank you, sir. Thank you, sir. Good afternoon to all of you. Today we are honored to hear from a veteran scientist from Rajiv Gandhi Center for Biotechnology on this occasion of 24th session of UL Space Club weekly webinar series. And Soniya ma'am gave such a great introduction about one of the most important topics, biotechnology, which is a game changer and will sculpt the future of human revolution. We are extremely thankful to Dr. Soniya for enlightening all of us with her knowledge. Thank you, ma'am. And it's my pleasure to thank every one of you at this very occasion of completing 24 weeks of this special program in this pandemic era. We are happy that not only students, but also many eminent personalities are always with us as a family in these webinars. And I thank all of those veterans for such a great cooperation. I thank all the participants, including students, teachers, and all others. Also, I thank our chief mentor, Sri E.K. Kutty sir, moderator Sajil sir, and student fellows of the Space Club, including Varun K, Master Bharat Srijit, Master Sriram D, etc. Thank you.
So my thanks are also there to Shajil sir, Kutty sir, and Padmavan sir, and Jairam sir. So thank you very much. Can I leave now?
Okay, thank you, ma'am. Great, thank you. Thank you.
Thank you, madam.
Okay, thank you, sir.
Thank you.
Okay, sir.
ശരി സർ, ഇപ്പോഴും കുട്ടികളുണ്ട്. കാരണം നമ്മള് നേരത്തെ
ലീവ് ചെയ്തോട്ടെ. കുട്ടി സാർ, നമുക്ക് പിന്നീട് ഒന്ന് സംസാരിക്കാൻ തോന്നുന്നു.
ആയിക്കോട്ടെ.
കാരണം ഐ വാണ്ട് ടു അറ്റൻഡ് വൺ മോർ തിങ് ആഫ്റ്റർ ദിസ്.
പപ്പൻ
ലീവായി.
ഓക്കെ
സർ, അപ്പൊ അത് പിന്നെ ഒരു ദിവസം നടത്താം അല്ലേ?